Our next speaker for today is Alan Alage, who will be presenting the Scilligence Dell platform. Alan has over 18 years of medicinal chemistry experience working as a bench chemist for several pharmaceutical companies. He joined Scilligence in 2019, where he is currently Associate Director of Sales and Marketing.
All right, give me 1 second while I share my screen. Let me see. There we go. Joanna, can you confirm that you guys can see my screen?
Yes, we can.
Awesome. Thank you. Well, thank you for the nice introduction, Joanna. And thank you, everybody, for joining us for our forum today. Today, I will be discussing Scilligence Dell platform, and I have the unenviable task of trying to condense about an hour and a half presentation into roughly 15 minutes. So I wish I was an auctioneer and I could speak fast, but if I gloss over some topics or aspects, my apologies. I can always get back to you with questions via email. So on the agenda for today is I will go through the Dell process overview, at least as we are familiar with it, based on experience from our clients. We'll describe some of the workflow challenges that we've seen and provide the data management challenges and the solutions that we offer with our Scilligence platform. And with that, then I'll introduce an overview of the Dell platform and try to cover as much as I can of the entire process with the remaining time. So one of the things that we've seen in working with different clients is that no one client does Dell the same way. Everybody has their own business rules and their own workflows that they like to modify in order to utilize their DNA encoded library process. And as we know, quick background on Dell's is it's essentially a very versatile tool to either beef up high throughput screening campaigns or in some cases replace expensive high throughput screening campaigns in an effort to find a lead or a hit that then can be optimized and then go through the drug discovery cycle that Jeff was discussing earlier in order to identify a development candidate for a drug. So what I'm showing here on the screen is what we know, or at least what we recognize as a generic Dell process overview. And it all starts with the library design. People get together and they want to design the type of libraries that can be done on DNA, which is very different from off DNA chemistry. They're going to procure building blocks, and they're essentially going to design the library on the building blocks that they're able to get from different vendors. Once the design is complete, then you actually get into the development and validation stage of Dell, in which you're now validating your building blocks. You're testing out different protocols for different cycles. You're looking at different DNA tags and maybe the codons and closing tags for the different steps of the libraries. Once you've accomplished the hurdle of gathering all of the tools necessary to initiate the library, then in earnest, you start the production of the library. And this can be a long process. This is not as straightforward as people read it to be. This involves a lot of QA, a lot of trial and error, and a lot of blood, sweat, and tears. After you've conducted your library screenings, you've made your libraries, you then want to do a binding, an affinity selection screen, where you'll select a target protein, throw your compounds at it, see what sticks, rinse out what doesn't, and then you have to enrich whatever is left over in the protein to try to identify the potential compounds that came from these libraries that you so meticulously worked in making in the past. And that's not even when it's over. You then have the unenviable task of trying to identify all of these enriched DNA tags that were amplified by a PCR, looking at the association to building blocks and codons, trying to weed out what could be junk or what can be desired structures. And then even after that, once you've identified something, it will not highlight a final compound. It'll simply give you a recipe for what could be a final compound, or perhaps a fragment, or perhaps a contaminant that is actually binding to your protein. Once you've gone through all of these steps, you then follow up with off DNA chemistry in an attempt to synthesize something that will hopefully bind to your biochemical assay. So this is a process this is as generic as we can highlight the process. So of course, there are numerous challenges to this workflow, right? It's very complex. There's a sheer volume of data that's generated just in one Dell library. And a common solution that we've seen clients use is a file system to keep track of all this data. This comprises of Excel spreadsheets, Word documents, email back and forths, and it's just not a sustainable system of managing this complex amount of data. Normally, what ends up happening is it just doesn't scale. It may work for one, perhaps two Dell library campaigns, but as you start to grow and as you start to get more sophisticated in your approach, you quickly see that it is not an efficient management system. So it leads to inefficiencies, human errors. It doesn't really allow for flexibility, traceability, just the overall reliability of the Dell workflow process. So we proposed a database database management system, and the difference is that this allows for consistency, integrity, and availability. Because all of your data is stored in a database, your team has access to it in real time. It's the most up-to-date data. It's not old. It doesn't get lost in emails or in Excel spreadsheets, and it's accessible 24/7. Here, you can manage all of your processes, right? Your building block validations, your DNA tags, the protocols that you've developed for various cycles. You can utilize Scilligence's built-in chem informatics and bioinformatics capabilities. So Jimbo and Jeff just discussed Helm and monomers to track your DNA tags, perhaps your headpiece, as well as the small molecule capabilities that I'll show you in the upcoming slides. If you do not have our suite of software modules, you can also integrate with your existing software via APIs or via direct database integration. And as always, we do provide configuration and customization for the client, because as I mentioned at the very beginning, no one company does Dell the same way. Everybody has their own unique twist on it. So these are the modules that we utilize for our Dell platform. Today, I'll discuss two of them: the Dell platform itself, which is essentially Regmal with some Dell add-ons, as well as ELN, which integrates with the Dell platform to help out in some of these processes. You also have access to inventory, SDMS, and PMF. With inventory, you can store your plates of building blocks, your DNA tags, any other consumables that you may need in this endeavor. SDMS is very useful for large data storage. We use it for two functions: large data storage that can be linked in directly through our Dell platform, and as well as integration with instrumentation. So oftentimes people use several liquid handlers to go through the production phase of a Dell library campaign. And in the end, PMF is a module that's a workflow engine that actually allows for all of this to integrate seamlessly or to a third-party instrumentation as well. But for today's talk, I'll mostly focus on the Dell platform. You'll hear me refer to it as Regmal or Regmal Dell and ELN. So what I'll mostly focus on is the first three chevrons of the process, which is our library drop-down menu on the Dell Regmal platform. We'll get through to the screening aspect once the libraries have been complete. And if time permits, I'll be able to show a few screenshots of how a user can utilize the selection data aspect of our Dell platform to try to enumerate the hits, to try to identify the highest scoring hits, and get the sequence counts that they need out of this entire process. Okay, so I'll start at the beginning, the library design. The Dell library, one of the first things you have to do is register the library. So when you first register your library, you'll have the ability to input a variety of different metadata that you will need. What I have on the screen here is just a library detail tab that has just a very generic synthesis path for a simple Dell library using cyanuric chloride. And you can see in the synthesis path, we have a potential headpiece with an amine linker that will be used to drive the synthesis path. And I know this is not a complete synthesis, this is just as quickly as I could sketch it for the presentation. With it, you can also identify the cycles that you're going to use in your Dell library. And it can be either 2, 3, perhaps even 4 cycle campaign that can store the reaction type for each cycle. And again, this is challenging on DNA chemistry that you have to use, as well as the building blocks that are associated, any DNA tags that you're going to use after every iteration of the cycle. All of that is stored in the Dell Regmol database. You'll also be able to register the closing tags that you'll use for this library, excuse me, for the library, as well as moving forward the selection samples that came out of the binding affinity campaign that you use for your protein binding screening. Once you've initiated a Dell, registered your Dell library, you have to buy the building blocks. And once you buy those building blocks, you need somewhere to register them and upload them. We're able to do that for you. You can register your purchased building blocks either one by one or in bulk. You'll have the capability of uploading those, and you'll even be able to peruse what you have in the Explore Building Block section, where you'll see the variety of building blocks that you've purchased in the past, what libraries they've been used in, have they been used up. If you have integration to inventory, you may even be able to see the location or the container where those building blocks are stored, but all of that is managed in the Dell Regmol side. Now you have 2 options for uploading validated building blocks, because after you've purchased the building blocks, of course, you have to validate it to make sure that they're actually going to work in your cycle. You can either directly upload validated building block data into the Dell Regmol module, and you would do that through this arrow here, and it would be a flat file that contains the data that you need to ensure that this is actually validated, or you can push it from our ELN site, and I'll show you what that looks like. If you do have access to, if you are going to upload it through our Regmol, there is a pop-up screen that will allow you to populate a lot of metadata for the building block, and you can identify the number of steps that you took to validate this, put in what you need to put in, and save it and upload this data. Of course, you can also do it in bulk, as I showed in the prior screenshot. We also, however, have integration with our ELN, and our ELN has a Dell add-on drop-down menu that has a variety of highly structured templates that you could use to help you with this Dell process. And the one that I'll highlight now is the BB validation template. So this template, as I said, it's similar to our single-step reaction template. For those of you who already use our ELN, will be a little bit familiar with it. However, this has been customized for building block validation. And in here, you'll be able to test your purchased plated building blocks with the reactants, run the validation step, and push the results into Regmol after you've completed your validation campaign. Okay, so that's just validating the building blocks. You also have DNA tags. Some clients like to validate their DNA tags or their codons in order to ensure fidelity. Others just trust the vendor of the building of the DNA tags. Either way is fine. There's a similar architecture for uploading DNA tags to building blocks. Again, you can either register your purchase tags in bulk or one by one, and you can explore in the DNA tag store what you have available in your inventory, what libraries have been associated with it, amounts, and a lot of other metadata that I will not get into complete detail for the sake of time, but you'll be able to view that in our Batch Explorer for the DNA tags. Okay, and of course it links into Scilligence inventory if you're using our inventory system. Okay, so you've bought the groceries. Now you actually have to get the recipe to make the meal. So now you have to create Dell protocols. Again, similar to the prior steps, you can either upload the protocol directly into Regmol or you can push it from ELN. In Regmol, you have the advantage, as I mentioned earlier, of capturing everything in one database instead of in different Excel sheets or Word documents. So all the protocols that you create can be stored in our Dell Regmol module. This is just a screenshot of what it would look like. I only have two protocols shown here, one that was directly uploaded and one that was pushed in from ELN. But you can see the details of the protocol, the name, the building block functional groups that were used, the DNA tag functional group. In this case, it's a carboxylic acid, it's a peptide coupling within a primary amine and a carboxylic acid, the buffers, the reagents that you use, etc., and then a generic scheme. Uploading it directly from Regmol utilizes our drop-down menu, so you can quickly populate commonly used building block groups, buffers, scaffolds, etc. Right here, what I have in the screenshot is just the reagent highlighted. When you double click on it, you'll have a selection of drop-down items to go through. These are populated in a dictionary. I will not get into that for this talk, but you can populate it and make it as large as you need to. And the idea here is to quickly create protocols by selecting the reagents and the options that you need. Oftentimes protocols are very similar with one minor tweak, either a change in buffer, maybe a change in temperature, etc. And so they're usually reused or used for different Dell libraries. You can do this quickly and at ease with Regmol. Also, you can highlight the synthesis path and maybe the steps for that procedure. That's directly to Regmol. If you choose to go the ELN route, again, as I mentioned earlier, we have an on-DNA chemistry highly structured template that allows you to do this and to push your protocol testing directly into Regmol. So this gives you the freedom to try different protocols and only push the ones that were successful into Regmol. So you don't have a population of protocols that just don't work. So once you've drawn your generic reaction scheme, added the reactants, added all the metadata that you want to in order to test this out, and the results are successful, you will then be able to push it into Regmol and it'll be populated with the data that you need. So in this screenshot, I have notebook 001 experiment number 3. Let's assume that this was a successful protocol test. I'm able to push it and link it into the Dell Regmol database. So if I were to click on the next screenshot, I scroll down a little bit so you can see, but this is protocol 2, and you'll notice that the experiment number that I pushed this from is captured in Regmol. We also redundantly decided to name it the notebook. That was necessary, but everything is linked in and captured in this database system. Okay. All right, you've got your groceries, you've got your recipe, now it's time to actually cook. Once you've validated everything and everything is in place, mise en place, now you can get cooking. The easiest way for a client to do this is to utilize our ELN and to just push everything in ELN. ELN has, again, highly structured experiment templates that can be used back and forth in cycles to populate your libraries or to actually do the production library. I'm going to start in the middle with production chemistry, which will be the on-DNA chemistry, followed by a ligation. And then at the end, the production after pool, this is when you'll introduce your closing tag in order to wrap up this library. So I'll very quickly go through these things. In this scenario, we just have on-DNA chemistry. We'll probably do a wine red amidation and get to this intermediate. The next one will ligate a new tag, tag 2 to tag 1, and repeat this as often as necessary for your 2, 3, 4 cycle Dell library. Once that's done, you'll go to the after pool experiment template and add your closing tag. And again, this is the closing tag that you probably already uploaded and registered into Regmol. So you can actually pull that closing tag into the ELN template if it's already been registered in the Dell Regmol series. We've completed this and you've done your library production. All right, I don't know how am I on time? I think I'm 18 minutes. I'll try to speed up. I won't go into too much detail in the next step for the sake of time. But once you've done and you've created your libraries, now the next step is to do an affinity selection screen, identify the hits that actually bound to your protein. And then I won't discuss follow-up after that because that's off DNA synthesis, and that would be another talk for using our ELN. But anyways, the key points for library screening is you've done your libraries. You can either do one library for an affinity selection campaign, or groups of libraries. Let's say you've done 5 Dell libraries and you want to combine it. We call this a super pool combination. And you want to throw all your libraries at this protein target to find out what sticks. You have the capability of doing that in our Regmol site. You can register the protein that you're going to use as your target sample for this binding affinity selection campaign for binding. You can create the affinity selection campaign and store it in Regmol. There you will define the protocols that you use to run this assay, any primers or closing tags that were utilized in the creation of these campaigns. Afterwards, excuse me, you can then go through the results of that campaign through our next generation sequencing capability. And I apologize, I don't have a screenshot for this as I'm going rather quickly, but you store that in Regmol. You can store any selection samples that you want, that you've identified from these campaign results, and associate them to the library that they came from. And then at the end, off our software using third-party tools, you can then upload the enumerated hits that you've identified, the sequence counts for NGS, or any aggregated scoring system that you utilize. All of that metadata can be uploaded directly into Regmol and captured so that you can do some preliminary searching for, oh, actually I do have something here, for enumerated hits. So these can be uploaded in bulk. You can search for any enumerated hits using our hit index search feature, and it allows you to quickly identify. This will not bring up the structures of what you've made. This will merely bring up the index so that you can later dig down and identify the potential recipe for the compound that you want to identify. And so here I just have sequence counts can be uploaded, as well as any aggregated scores that you will then conduct outside of our Scilligence module. I think I've spoken for 21 minutes, so I'll end it here. To summarize, we have a platform that encompasses as much as we can for the DEL process that can be configured or customized for the client need. And we have clients that are using our platform as we see. So Wuxi AppTech is probably our largest client that's utilized a highly customized version of this DEL platform. I'll stop now. I thank you for your time. And I don't know if we have time for questions. I don't want to cut into the next speaker's time.
Thank you very much, Alan, for your presentation. As everybody can see, there's a lot to cover. If you do have questions about the DEL platform, again, please email us at info@scilligence.com.